Good evening, everyone. Am I audible? Please confirm if my screen is visible and I'm audible to you all. Okay, so today we'll be starting with the Unit 5C questions. We'll be discussing the Unit 5C questions only, which is organogenesis. We have already seen few of the topics in the previous Brahmastra series of Unit 5, developmental biology. Today we'll be seeing a few more. The first question is from September 2022. It is on mammalian development. Okay. Now, last class I had mentioned to you that there are some specific factors which are expressed in the mammalian embryo when it is in the blastula form called as blastocyst. Now, in mammalian blastocyst, you have some inner cell mass cells. Okay. And then there are some cells which are present on the periphery. So, these peripheral cells, these are what are trophoblasts, and the inner cell mass they give rise to the embryonic structure, whereas trophoblasts they mainly give rise to placenta, extraembryonic membranes. So few of these statements are made regarding the factors that make these cells undergo the function that they are supposed to. Which one of the following option represents the correct combination of statements? Yes. Tell me if anyone has the answer for this. So this is an important topic, mammalian development. We see some questions regarding their embryology. Okay, you also get from this area as well. Yes, any guesses for this answer? Okay, Priyanka says one. Let's have a look. Now see, zygote is a totipotent stem cell, right? First of all, potency, what does it mean? It means the capacity to form a certain cell type. And there are different levels of potency. Okay, Hardik says it's 3. Okay, let's have a look. You have completely different statements, A and B and C and D. Okay. So totipotent is something which can generate the entire embryo. Okay. All the cells of the body they can form. That's what is totipotent. Pluripotent, on the other hand, means they can only form certain cell types. Not all cells of the body, only certain cell types they can form. Okay. Then we have 3 regulatory transcription factors which are OCT4, NANOG and SOX2. They are present in the inner cell mass and they help maintain the pluripotency of inner cell mass. Okay. So if anyone would know that yes, OCT4 in the inner cell mass, OCT4 initially is present in all the cells of the zygote, okay, but later it only gets restricted to the inner cell mass. The trophoblast doesn't have any of the OCT4. Why is it so? Because trophoblast cells start expressing another factor which is called as CDX2. Okay, and CDX2 is responsible for inhibiting OCT4. So this sign is for inhibition. CDX2 will be inhibiting OCT4, and that is why you don't have OCT4 in this trophoblast. It is restricted only in the inner cell mass. Okay, so clearly D is wrong. So neither 3 and 4 can be correct. Why? Because it's saying CDX2 upregulates OCT4, but it is actually the other way around. It downregulates OCT4. Okay, so the correct answer over here is option 2. As I said, inner cell mass they are pluripotent because they cannot make the placenta. They can only make the embryo, most of the cells, but they cannot make the extra embryonic membrane. That is why D is correct. You have to choose the correct combination. And even the 3 regulatory factors, even this is correct. Okay. I said nanoxox2, they are also present in inner cell mass, and they maintain the inner cell mass. You would know it consists of embryonic stem cells, which are pluripotent in nature. So these are the factors which help in maintaining them. All right. Now, why this is wrong? The cells of inner cell mass. Why this is wrong? Because it is mentioned that zygote is a totipotent stem cell. See, zygote can be totipotent, but zygote being a stem cell is a question mark. So that is why A hasn't been considered as correct, right? Zygote has the totipotency property as it reserves the capability of forming embryonic and extraembryonic membranes. However, it is not a totipotent stem cell. So, what is a stem cell? A cell that renews itself along with the formation of one daughter cell, right? So, when you have the zygote will divide into two cells, right? So, it's not possible that out of these two cells, one of the cell could make the zygote again. That is not possible, right? If the zygote can make another zygote like it, then only it will be called as stem cell. But the zygote is not stem cell. It only goes on dividing and forming the cell fates in the forward direction. So you have to be very critical when it comes to the keywords that they use in the statements of the question. Okay. And that's why it's suggested that before the exam you should not prepare intensely. Otherwise, your brain is not ready to process this kind of critical words or statements. So, totipotent is correct, and probably will mark A as correct because of totipotent, but you should analyze each and every statement. Okay. So, yes, Sarita, that's right. Option 2 is right. Now, next question is from June 2019. This is from limb development. As I mentioned, there are few topics, scattered topics in Unit 5C Organogenesis. One of them is this mammalian embryonic development. Second is this limb development, neurulation, neural tube formation, then sex determination, vulva development. We have already discussed questions about it in last class. And also you have this camp aggregation. Then small topics like islands induction and regeneration is also there. But till date, the metamorphosis part and environmental control of sex regulation, it has not been asked in questions yet. But you never know, there can always be a first time. Okay. All right. So coming to this limb development, during wing development in chick birds, ectodermal ridge is removed. If apical ectodermal ridge is removed. So what is this AER? You should have a knowledge to solve this question. The limb development ceases. Okay. If you remove this, it has been seen that the limb development stops. On the other hand, if you place a leg mesenchyme directly beneath the wing AER, instead of removing it, if you place the mesenchymes are loose cells which are not connected in a layer, though they can form any kind of structure. So ideally, they accumulate under the limb bud and they convert into or they differentiate into the muscle cells, cartilage cells to extend the limb as it comes out of the body. Okay. So if you're trying to place the leg mesenchyme under the wing AER. Okay. So in that case, distal hind limb structures, they will develop at the end of the wing. And if limb mesenchyme is replaced by non-limb mesenchyme beneath AER, then it will regress. The limb will stop progressing any further. So this may demonstrate these few options. So you have to find out based on this experiment, which out of this above statements are demonstrated. Okay, it need not be that which statements are correct, but based on this experiment you have to conclude that which of this above demonstrations would be correct. So which combination of above statement is demonstrated? Yes. Do we have any guesses here? None? No one? Okay. So Sarita says option 2. Okay, we'll see. Yes, Priyanka also says option 2. All right. So see, the limb mesenchyme cells, they induce, and actually this question is based on an experiment. So yes, your answers are correct, both of you. So this is the experiment that it is based on. You have a four-limb mesoderm, which is having this AER. Okay, AER is what? Apical ectodermal ridge. And what is this AER? Why it is required? So you will see if I zoom out this to you. Okay, so you will see that there is some elevated region of the outer ectoderm, and below that you have some clump of cells, a group of cells. So these are what are the mesoderm. Okay, mesenchymal cells. Yes, very good, Akhilesh, that is the right answer. So we have the mesenchymal cells or the four-limb mesoderm, which is extending the limb out. So both are required. AER gives signal to the mesoderm or mesenchyme, underlying mesenchyme cells to extend out, and mesenchyme gives signal to the AER to maintain, to keep it maintained at the tipmost part of this limb bud. Okay, if you see, when the limb grows normally, so we don't have a diagram here. Okay, no worries. So if the limb grows as and when the limb will grow here, okay, so along with that AER will also migrate. If the limb grows more, the AER will migrate. So always the AER will be present at the tipmost area because the limb will be moving from proximal to distal growth. The proximal to distal growth and differentiation of the limb bud is made possible by a series of interactions between the limb bud mesenchyme and also the AER. So it's kind of a reciprocal interaction. Although the mesenchymal cells induce and sustain the AER and determine the type of limb that is to be formed, the AER is responsible for the sustained outgrowth and development of limb. AER is very important, then only it can give the signals to the underlying mesenchyme to be maintained in a proliferative phase. They have to be proliferating, otherwise it's not possible. Okay. The AER keeps the mesenchyme cells directly beneath it in a state of mitotic proliferation and prevents them from forming the cartilage. See, the limb should continuously extend out. That is why the mesenchyme has to be kept in a proliferating stage. If they are turning into cartilage, then the limbs cannot they cannot extend out anymore. Okay, they should be in an actively mitotic state. So obviously we have those experiments which is made. AER is removed means without AER, the underlying mesenchyme cannot be in proliferative state, so limb development stops. It doesn't continue. If you put some extra AER, then as and when it extends out at the distal end, that is the tip area, two different directions are formed. Like wing is duplicated if you have extra AER. Okay, because the mesenchymal cells are more active here. So because you have extra AER, they will extend out further in the distal end of the limb. If you place a different mesenchyme, instead of wing you put leg, then you will see that from the proximal end of the body, proximal means closest to the body, the limb starts out as wing, but at the distal ends, this leg mesoderm doesn't get any more signal from the body, so it continues to make what it was supposed to make, that is leg. But if you take non-limb mesoderm, then AER is not able to keep maintained. Okay, as I said, the limb mesenchyme is supposed to maintain AER. So if you keep non-limb mesenchyme, then AER will stop progressing. And if you replace the AER with a FGF bead, FGF, why? Because ultimately AER will release FGF. Okay, fibroblast growth factor is what is necessary for this limb development. And if you replace it with FGF, then it can again make AER. In that case, normal wing will proceed. So that's why here, definitely the limb mesenchyme cells, they are able to maintain the AER, induce and sustain or maintain the AER. So A is correct. Mesenchymal cells specify the type, wing or limb. So that's why when you took the leg mesenchyme, in the distal ends, it was forming the like limb structures will be coming at the end of the leg structures will be coming at the end of the wing. So that's why mesenchymal cells will specify whether it will form forelimb or hind limb. But AER is not responsible for specifying the type of limb, whether it is wing or leg. Okay, so AER is not responsible, but yes, AER is responsible for continued growth and development of the limb. Okay, and AER does not specify the type, whether it is wing or limb. AER does not specify. So these two are opposite statements, anyways. So C is wrong and E is correct. So that's why correct option is option 2. All right. Next is another question on limb development, but this is a little bit more detailed one. As the limb grows outward, so there are three regions of the limb, as you would know: zygopod, stylopod, and autopod. And you should be knowing which area is where. For example, from the shoulder area till the elbow, then from elbow to the wrist area, and then the finger area. So what is which region you should be knowing to solve this question, because accordingly you should know, you know that the limb always extends in a proximal to distal direction from the body. Proximal means closer and distal means further away. So obviously the shoulder part will form first. So which is the shoulder part you should know, and each phase is characterized by a specific pattern of Hox expression. So, what are Hox genes? They are reserved sequences, they are conserved sequences throughout many organisms. And most importantly, they are a group of transcription factors which can control the genes of organogenesis. Okay, ultimately, what structure has to form in a certain region of the limb? Let's take the fingers. Okay, so what kind of transcription factor is required? So there are some specific Hox genes which are exclusively expressed in only the regions where they're supposed to form that organ, right? So this is one statement. Second is zone of polarizing activity. It is ZPA. This region, it's a region present in the limb bud, and as the limb extends, the ZPA also continues with the limb outgrowth. So the ZPA has to be maintained. If it is present in the limb bud initially, then it cannot be just present here. As in when the limb extends, the ZPA also has to keep maintained. Like in the further areas of the limb, the ZPA has to keep on continuing. So that is maintained by the interaction of the FGFs from the AER and SHH, which are expressed from the mesenchyme. So AER releases the SHH and also the FGF, so that maintains the ZPA. Although cell death in the limb is necessary for formation of digits and joints, it is never mediated by BMPs, which is only responsible for differentiating mesenchyme cells into cartilage. So you should understand that when a limb proceeds, under the AER, you have actively dividing mesenchymal cells, but in the proximal areas from where the AER has already crossed, it has gone further ahead. In the previous areas, whatever cells have been laid down, they should form the cartilage, okay, because they are not extending any further. So that is done by BMP according to this statement. They are responsible for differentiating them into cartilage, but they are not involved in the cell death in the limb, which is necessary for digit and joint. So joints means there also we need the formation of the cartilage, and in the digits also, in the fingers you have these digits, right? So here like this digits are there. So it's saying that BMP is not involved. So think about it whether this is true or false. The dorsal ventral axis is formed in part by the expression of W27A. So we have the anterior, posterior, sorry, this we have the proximal distal axis question. Statement was one is on proximal distal, that what forms first, right? Then this one is for the this is for the anterior posterior. ZPA is regarding the anterior posterior axis. And the fourth statement they have asked regarding the dorsal ventral. So the back side of your hand, where the knuckles are there with which you can hit someone when you make a fist. So that side is the dorsal part, the back side of the hand, and the front side of the hand is the ventral part. Ventral means where the palm lines are there, right? So this question is basically on the entire axis formation of the limb, okay? And this is regarding the signaling. So which statement is not correct, they have asked. Yes, so Lokesh says option 3. Anyone has any other guesses? Yes. So yes, Lokesh, your answer is right. It is option 3. So as I mentioned, you have this autopod, stylopod, zeugopod, and autopod. You can see this is the body axis, and from here the limb is extending. The part closest to the body is the stylopod. So obviously that part will form first because the limb extends in the proximal to distal area. Okay, proximal areas form first and distal areas at the end. So after stylopod, you have zeugopod, and then the end areas, those are called as autopod. So as the limb grows outward, the stylopod forms first, then the zeugopod, and then the autopod is formed last. Each of these three phases involve the expression of Hox genes. So first statement is correct. So what are Hox genes? So here you can see this is the proximal to distal. What are Hox genes? So you have different Hox D sequence starting from 9 to 13. So Hox D9 will be expressed in the stylopod area. Then 10 and 11 will be in this area. Then 12, 13 will be in the autopod area. So something like that. Each of the Hox is responsible for forming specific structures, right? Then we have the ZPA, which is the anterior to posterior axis formation, because obviously if you're talking about a limb outgrowth, then the top part is anterior and the bottom part is posterior. Okay, so here also this elevation at the top, it is AER, and under that you have mesenchymal cells. As I said, the limb as it continues to grow, you have something which is called as ZPA. So you can see ZPA, this is the limb bud, this is the anterior top side. It is not present in the anterior part. ZPA is present in the posterior part. Okay. And the AER, it's given in red. And under the AER is the mesenchymal cells, active mesenchymal cells, which is called as a progress zone because they are progressing forward, right? So AER is giving signal to the ZPA, and ZPA is giving signal to the AER. So they maintain each other. That is why as the limb extends out, the AER migrates, and along with that the ZPA also migrates, because you want the anterior posterior axis formation in the entire limb, right? You don't only want at the proximal area. Even in the digits also, if you put out your hand, your thumb will point towards the top and your pinky finger will be down. So that means at the tipmost end of the limb, if you take your forearm, that is the hand, there also you should need the ZPA. That's how they will maintain the anterior posterior. Otherwise, your thumb will come out any direction, isn't it? So ZPA is established and maintained by the interaction of FGF8 from the AER. So you can see that FGF8, which is blue color, it is released from the AER. It is given to the ZPA region. And mesenchyme made competent to express sonic hedgehog, which is SHH. So ZPA is expressing the SHH and it is giving the signal to maintain the AER. Both are maintaining each other, right? Then coming to the fourth statement, as I said, it is regarding the dorsal ventral axis. So this is responsible, the molecule responsible for forming this is WNT7A. It is in the dorsal portion of the limb ectoderm. So WNT7A maintains the expression of SHH in the ZP and FGF4 in the posterior AER. So yes, whichever side WNT7A signaling is there, that side is dorsal, and whichever side BMP signaling will be there, that side will be the ventral part. Okay. Now, cell death in the limb is necessary for the formation of digits and joints. Otherwise, the sculpting of the fingers and digits and everything will not happen properly. This is mediated by the bone morphogenetic protein. So, yes, option 3 is not correct because it is mentioned that BMP doesn't are not involved in that process. Okay, so effect of BMP can be regulated by the noggin protein, and the BMPs can be involved both in inducing apoptosis and in differentiating the mesenchymal cells into cartilage. Okay, so that's why option 3 is wrong. All right. So just want to announce some workshops and internships. Tomorrow, 1st March, 10 a.m. onwards, we have a very important workshop. It's a national workshop on big data biology, and we have eminent speakers who would be joining the session. Um, Dr. Subba Reddy, who is a postdoctoral researcher at TU Berlin, Germany, and Dr. Shruti Desai, who is an omics data expert, along with Dr. Nilofer Sheikh, are in-house bioinformatics faculty member expert, and along with me. So AI, genomics, and the future of precision medicine. We will be. It's a one-day workshop. We'll be learning about it. If you want to enroll in this, or if you want to join the workshop, then you can call on this toll-free number or the office number, or else you can directly go in stores.biotecnika.org. Okay. And along with that, 4th March onwards, we are also starting a 120-day hands-on internship on data science for biologists. As you know, these days, AI, ML, then data science, bioinformatics, these are very hot topics. Okay, if you are going into research and you don't have an idea about these things, then you might feel that something is missing. Okay, because in almost every research now there will be utilization of these concepts, and we have again experts of this topic, Dr. Nilofer Sheikh, Mr. Pradut Banerjee, and Dr. Ilamati. So they have their own specialities like bioinformatics or CAD or AI/ML, and this will be online along with offline project also. But keep in mind, there are limited seats for it. Okay, so if you want to be a part of it, then you have to enroll yourself now. Otherwise, later the vacancy might get filled up. Okay. Then the next question, it's again on the limb development only, some things which we just learned. So maybe you can try this on your own. Given below are statements which are related to the expression of the pattern following genes in vertebrate limb development. So in June 2021, there was surprisingly many questions on limb development. Total 3 questions were there on limb development in part C. I think morning and evening batch combined. So there were some 3 part C questions on limb development. Okay, so that is very unusual. Probably this is not unusual in other question papers also, because depending on the question paper setter, they will be whatever is their specialization. Accordingly, they will give more questions on that topic. Okay, so that is on your pure luck. We cannot really predict these things. Yes. So the statements are: limb 1B gene is expressed in dorsal mesenchyme, SHH is expressed in the posterior region. So just now we saw where SHH is expressed, okay? And you should be knowing what is this LMX1B gene. And WNT7A, just now we saw whether it is expressed in dorsal ectoderm or not. So that also you should know. And HOX A13, HOX D13, they are expressed in the distal region. Just now I said you have some HOX genes, that is HOX D9 to 13. That means 13 will be at the most distal region, right? TBX5 and FGF10 are expressed in the lateral plate mesoderm, involved in the formation of limb bud. So the question is something different. They are not asking which is true or false. The question is the nail patella syndrome in human and the syndrome in mouse, which exhibits foot pads on both dorsal and ventral surface of limb. See, foot pads, that means they should be present on the dorsal side, but it is seen present on both dorsal and ventral, because of which you have the nail patella syndrome. Okay, they are associated with which of the above mentioned gene functions? So whatever it is, definitely C will be there, because it is talking about the dorsal ectoderm, right? So since dorsal side is duplicated in the ventral side also, so clearly C will be a part of it. So if C is a part of it, then the only option that you have over here is A and C, right? Because this is clearly the posterior region. So SHH won't be involved in the dorsal ventral axis. Even HOX D and 13 is the proximal distal axis. This is anterior posterior and this is proximal distal. Then TBX5 and TBX4, they are necessary for specifying a certain limb type. Like TBX4 will be specifying forelimb, whereas TBX5 will be specifying for forelimb and TBX4 will be specifying for hind limb. So that is the limb type. It has got nothing to do with the dorsal ventral. But LMX1B gene is expressed in dorsal mesenchyme. So that is also correct. So you can see in the previous question also I was showing you. So we have the at the AER tip. So FGF8 is released, and wherever FGF8 is there, the concentration wherever it decreases, there we have the SHH signaling. Okay, and how does WNT7A give the dorsal signaling? It will be releasing this LMX1B. It is a transcription factor only, the limb domain homeodomain transcription factor. So this is what is responsible for limb dorsalization. In the limb, LMX1B is induced by WNT7A from the dorsal ectoderm. They are present in ectoderm area only, and its expression is restricted to the dorsal mesoderm only. Okay, that is limb 1xb won't be expressed in ectoderm, it will be expressed in the mesoderm area under the ectoderm, right? So loss of 1xb gene function in mice results in loss of dorsal autopod. Autopod means the end region of the limb where the fingers and all will be formed. So see, these are the Hox genes, right? So different Hox genes are expressed. Hox 9 will be expressed only in the stylopod. Then Hox 10 will be expressed in stylopod and in zygopod. 11 will be expressed in zygopod and in autopod, that is the fingers. And zygopod is the radius and ulna, and stylopod is the humerus. We are talking about the fourth limb here. And 12 and 13 will be expressed exclusively in the autopod, right? So here, if LMX gene 1 is not there, then the dorsal autopod and zygopod patterning, it will be lost, right? Patterning will not happen properly. The autopods have a symmetrical ventral phenotype with dorsal foot pads. So because of which nails also don't form. The dorsal side, it doesn't form properly. So the dorsal hair follicle is also not there. At the dorsal side, we have the hair follicles. So we are talking of the limb 1xb if there is a mutation, right? So that's why beside the limb, mice lacking functional LMX1B exhibit abnormal eye, cerebral and kidney development, which accounts for the prenatal lethality. Even before they are formed into adults, they will die. Okay. In contrast to mice, single allele variation in human that disrupt this LMX1B will cause the nail patella syndrome. Right. Okay. Next question again on limb development. So as I said in June 2021, so which of the following state combination of statement is correct? Yes, are you people following or not? I have not received any answer for the previous questions. Yes, I think previous two questions I haven't received any answer. What is this? Only Lokesh has answered. Yes, try to answer everyone. Come on. Hardik, Priyanka, Sarita, Akhilesh, Lokesh. Try to answer. See, you have this list of the components which you will find in limb development, and these are the explanations of what exactly those are. So this is very easy. You know, in match the following, if you know confidently at least one or two matches also, the rest of them will not be much difficult for you. Come on, can you do it? So what is AER? At least you will know what is AER, right? Apical ectodermal ridge. Am I even audible to you? Come on, students. If you answer, then even if it is wrong, once I correct it, it will remain with you until your exam. Okay, you will not forget easily. So you all are writing 1st March exam? Yes, all of you are writing, right? Yes. So start with the part C questions. That ideally is the norm, because your mind will be fresh, and if anyone is traveling from a long distance to the center, then you should leave early, because immediately from the traffic, when you directly go to the exam hall, your mind is in full commotion, and writing a full 3-hour paper, it will be difficult for you, right? So your performance depends a lot on these small factors as well. Start very early, reach the center at least 1 hour before, sit there, have some light snacks, and wait for your turn. That way your mind will be little bit in a calm situation. And these part C questions, they require a calm and concentrated mind, right? Okay. So see, AER is nothing but, as we saw, um, it is the thickening of the ectoderm just at the tip of the limb. So obviously AER will be option 2. So C is option 2, means any of these can be correct, right? So then we can look at the progress zone probably, okay? Or else autopod, you have this autopod. We know the fingers are called as the autopod. So clearly this is F will be 6. It is the distal part of the tetrapod limb. So F will be 6. Option 1 is the right answer. So see, just with only two things, we could, if we could be we were able to solve this, match the following question. What happened? Others haven't studied this part. See, don't leave limb development. Okay, you have tomorrow, today, tomorrow, whatever you can, you study from it, because unit C, this neurulation and everything, whatever you can, little bit you can study from this video only. You can rewatch it again. Many concepts I have given it here. So EMT, it is the epithelial to mesenchymal transition. Epithelial cells are the ones which are present in a layer. The cells are present in a layer. Mesenchymal cells are loose cells. Okay, they have come out of the layer and they can go and um, be present into any of the areas. They can get shifted or transported, right? So these are epithelial cells. They making up the mesoderm of the early somatoplure, undergo this transition and get included in the mesenchymal cell pool. From epithelial, they become mesenchymal, and then only they are able to accumulate under the limb bud. That is EMT. Mesenchyme. So these are loosely organized, mainly mesodermal embryonic tissue. So they are present just under the AER, mesenchyme cell. Loosely organized means they are not connected in a layer of tissue. They are just loose cells which can accumulate. Okay. Then we have AER, I already mentioned. Progress zone. So it's a proliferative mesenchyme. AER maintains that mesenchyme in the mitotic phase or the proliferative phase. That only will extend the limb further. And ZPA, which we just saw, it is found within the most posterior region of the limb bud, because it maintains the anterior to, it not maintains, it actually determines the anterior to posterior specification. Okay. So the explanation is also given regarding the ZPA and normal development. So you can see here, right? All right. Next question is regarding neurulation. One question, again from June 2021. Given below are some of the statements in connection with neural tube formation in vertebrates. I have mentioned that in development biology, most of the times you will get, many of the times you will be getting comparison between different processes in different organisms. So maybe the neurulation, gastrulation in amphibians or chicks, or the fertilization in sea urchin and mammals. So such kind of differences here you have also got between birds and mammals, neurulation. So the question is based on primary neurulation. The cells in primary neurulation, the cells surrounding the neural plate. Yes, Priyanka, that's right. You had answered. I just saw it now. Yes, you are right for the previous question. Very good. Yes. So, in primary neurulation, the cells surrounding the neural plate, they will direct the neural plate cells to proliferate, invaginate, and separate from the surface ectoderm to form an underlying hollow tube. So, what is the neural tube? Obviously, it is part of the nervous system, central nervous system, peripheral nervous system will be also formed. But this neural tube, it will be um, formed in two parts, one by two ways. One is primary and secondary. So in secondary, the neural tube arises from the aggregation of mesenchymal cells into a solid cord that subsequently forms cavities to create a hollow tube. So different parts of the neural tube, from the anterior portion to posterior portion, till a certain portion it will be by primary neurulation, and the rest of the portion will be by secondary. So that's what they have asked here. In birds, primary neurulation generates the neural tube from anterior up to the hind limb developing region. So in the vertebrate column, wherever the hind limb starts, so till that portion it is primary, beyond that will be secondary. In D, they are saying the in mammals, secondary neurulation begins at the level of sacral vertebrae. So you should have a knowledge of the different vertebrae also when it comes to this limb development or neurulation, because they can ask from which vertebrae the limb is generating, which and because the Hox genes are also present in anterior to posterior. If you consider in the vertebrate, there are some Hox genes because of which the vertebrae are different. You would see that till a certain point the vertebrates are joined. That's because of a Hox gene. After that, the vertebrates are not joined. Okay, I mean the bones that come out from the vertebrae, isn't it? The rib bones which you see. So that those knowledge also you should be having. Then anencephaly results when a failure to close the neural tube occurs, resulting in the forebrain remaining in contact with amniotic fluid. So which out of these are correct statements? Yes, any guesses? Anyone? Has anyone studied neurulation? Have you prepared it for your exam? No? Okay, no problem, Priyanka. I will explain it over here. Anyone else? Hardik, Sarita, Akhilesh, Lokesh, anyone of you want to guess regarding this question? Okay. So I'll have a look over here. Here the correct option is option 1. So there are two ways of forming a neural tube. In primary neurulation, the cells which surround the, so this is primary neurulation. We have the epithelium or the outer layer, and underneath we have the mesenchymal cells. Okay, the dispersed mesenchyme is there. So the primary neurulation begins with columnarization. That means whatever cells are cuboidal, they start becoming elongated like columnar cells like this. Slowly they will start to become rolled, or folding will appear over here, and the shape of the cell changes. See, if a cell shape is like this, so here you are getting a cell shape like this, okay? But if it changes to something like this, okay, like initially it was like this. If you see here, the cell shapes were like this, but if changes like this means they start folding overall as a sheet, they start changing the surface area, right? And at a point they pinch off from the outer epithelia completely, and that's how they form a neural tube. So primary neurulation, most importantly, it is derived from the ectodermal cells, because epithelia cells are derived from ectodermal cells. That's why it's colored as blue, as you can see. And this will be a vertebrate column. So just dorsal to the vertebrate column is the neural tube, and then you have the dorsal epithelium. Okay, so in mice, as you can see, in mammals, you can consider. So this is the transition zone. Before that, you have the primary neural tube. So in secondary neurulation in human embryos also starts below the level of the 32nd to 34th somites, which corresponds to the future third to fifth sacral vertebrae. So that is also correct. This is the sacral vertebrae, secondary nucleation from where it will start. And somites are some cartilaginous tissues which are present just uh, on the sides of the vertebrate column. So from the somites only cells will be released to form certain parts of the limb or even the other tissues, right? Secondary mesenchyme, if you see secondary neurulation, it forms from the mesenchymal cells. Now, mesenchymal cell mostly it will be part of mesoderm only, and that generally we mark it as red color in standard development biology books. So, first it starts with the condensation of the mesenchymal cells. Initially, they are dispersed, but later they start condensing like this. Then, a hollow inside some hollow opening will start beginning up. So you can see epithelial transition or cavitation takes place, because of which the neural tube is formed. So in secondary neurulation, the neural tube arises from a solid cord of cells that sinks into the embryo, and subsequently it hollows out to form the tube. So this is derived from the mesoderm, right? So in birds, the anterior portions of the neural tube are constructed by primary, that's right, while the neural tube caudal means towards the posterior side, 27th somite pair, that is everything posterior to the hind limb, that is made by secondary nucleation. So both C and D are correct statements. And even E is correct, which is referring to a condition called as anencephaly, failure to close the anterior neural tube. If the neural tube is not closed, it gives rise to the CNS, central nervous system. So if it is not closed properly, then it is obviously a lethal condition in which the embryo cannot survive. Here, the forebrain remains in contact with the amniotic fluid, and subsequently it will degenerate. Okay, so fetal forebrain development ceases and the vault of the skull also fails to form. So that is why here all the statements are correct. So A, B, C, D, and E, option 1, all the statements are correct here. Okay. Okay, then we have one more question which is based on sex determination. We had seen last class, right? So this anyone can guess. Following statements were made about the sex determination in Drosophila melanogaster. It is achieved by a balance of female determinants on the X chromosome and male determinants on the autosomes. So if you would know how the Drosophila sex development takes place, so does it develop by this X by A ratio, yes or not? You have to mark the combination with all the correct statements. Then we have a Drosophila with 0.66 value of X by A ratio would develop the would develop intersex type. So 0.66, that means you should know X by A ratio. What will be the ratios by which male and female specification will happen? And if you have 0.66, then what will happen? Okay. Due to the non-involvement of Y chromosome in sex determination process, we don't have Y chromosome in Drosophila. So, in case you have exodrosophila, then it develops as a normal fertile male. So, is that statement correct? Will those exo develop into male? And if they develop into male, will they be normal, sterile, or fertile? That you should know. High value of XA ratio facilitates the activation of the feminizing switch gene, which is called as sex lethal. Okay. So if you have high X to A ratio means X will be more than A. If you know in insects, the X by X and A ratio, the number of X chromosome and autosomes are not fixed in all the individuals. Okay. So there might be 2 and 2 or 2 and 3, 4 and 4. Like that, there can be some variety. And accordingly, what ratio will be there, it will facilitate activation of the feminizing switch gene. Switch gene, it is called sex lethal because it can be switched on only in the female phenotype. Okay, it won't be there in males. And sex specific expression of SXL causes selective activation of dosage compensation genes in female Drosophila. So if you have SXL, then it's a sex specific expression because, as I said, it is expressed only in female, not in male, because of which it will cause selective activation or dosage compensation in female Drosophila. So is this really correct? Okay. So yes, A seems correct. B also seems correct, right? And E is doubtful. C is doubtful. So ABD option 2 seems the right answer. If you would know something about the maintenance of SXL function, SXL is the sex lethal. This is the pre-RNA. Actually, entire sex determination process of Drosophila, it depends on the alternate splicing. So same RNA, but different proteins can be formed based on splicing. Okay. So here we have the pre-RNA of sex lethal 1, 2, 3, 4, 5, 6, 7, 8. But 3 has the exon, male specific exon, and 3 has a stop codon next to it. So what happens in female specific splicing, that is why it is sex specific sex lethal protein. So this third exon is completely removed. It is not expressed. From the second to fourth, intron along with the male specific exon is removed during the splicing of female specific specification. Whereas the default splicing, if it is not going for female specific, the default splicing is that all of these exons will be expressed. And since the translation, when it starts taking place, since it will encounter the stop codon, that is why you will not get the protein. The functional protein will not be there. That's why in male you don't have SXL. The male specific is the default splicing, which leads to a stop codon, and that's why no functional SXL is formed. Whereas the female, they will form a functional SXL, sex lethal, because they remove the male specific exon. Now, sex lethal will activate the transformer alternate splicing. Again, there are 4 exons. The default splicing is where all the 4 exons are present in the processed mRNA. And since this has a stop codon, again here you won't have any protein. But if SXL is there, then this goes into an alternate splicing where the second exon is completely removed. So that's why no stop codon and transformer protein is formed. Now TRA2 will be present in both male and female, But TRA2 should form a complex with transformer protein in order to splice out the double sex in a sex specific manner. Okay. Male doesn't have TRA2, male doesn't have transformer synthesized. So even if TRA2 is there, it cannot form a complex. And that is why the double sex goes for a male specific splicing. And if transformer is synthesized in females, along with TRA2, they form a complex and they will go for female specific splicing. Female specific splicing is only the first four exons, so that leads to a female double sex. And male specific double sex splicing is where the fourth exon is removed. So you have 1, 2, 3, 5, and 6. That is male specific double sex. Okay. So option is answer 2, as I said, because in Drosophila, sex determination is achieved by a balance of female determinants on the X chromosome and the male determinants on the autosome. X by A ratio should be there. I don't know if it is present. No, X by A ratio. So if the X by A ratio is more than 1, if it is equal to 1, then it goes for female phenotype. Okay. And if it is zero point 0.5, that means what? Autosomes are more than X chromosome. So basically, X chromosomes have the feminizing factor. That is why when they are present equal to each other, the X chromosomes are able to exert their effect because they will be having the expression of SX cell, right? But if the X chromosomes are less, autosomes are more, then they don't allow the synthesis of SX cell. So if SX cell is not there, TRA will also be not there. In that case, the specification will go for male specific character. Okay. So normally flies have either one or two X chromosome and two set of autosomes. If there is one X chromosome and two autosomes in a diploid cell, so the ratio will be 1 by 2. In that case, the fly will be male. But if there is two X chromosomes and two autosomes, then the ratio is 1, right? So in that case, it will be female. So if you have XO, that means Y chromosome is not there, then it will be male only in that case. Okay, but they will be sterile. So C is wrong as it is saying fertile male, right? So C will be wrong. And the first phase of Drosophila sex determination, it involves reading the X to A ratio. If you have high values of X to A ratio, they are responsible for activating the feminizing switch gene, sex lethal. So D is correct because it activates the SX cell in a female form. And if you have 0.66, then also it will lead to intersex because it is somewhere in between the ratio of 1 and 0.5. So 0.66 will lead to intersex, because of which B will also be correct. But then again, E is wrong because when you have the MSL loci, this is the MSL loci, and SXL controls this MSL loci. So you can see SXL is binding with the MSL loci. Okay. If that is the case in females, it happens, SXL is there in females, there will be no dosage compensation in females. Okay. But in males, you will have the dosage compensation. So they will regulate the dosage compensation, but not of the females, they will regulate it for the male X chromosome. So E is wrong because it is saying female Drosophila. Okay. So just want to remind you regarding the big data biology workshop, which will be happening tomorrow. You can still enroll in that. You just have to go to stores and register yourself. And also you can make a call in the toll free number. And apart from that, you also have a very important internship which is starting. It will be online, plus along with that offline project also will be there, hands-on training with some months project work. And this is data science for biologists. So if you're interested, you can enroll right now because for this one seats are limited, right? All right, so this is one more question. uh, I'll skip this as of now. Yeah, so let's have a look at this particular question. Okay, so in a hypothetical organism, this is from the recent June 2024, at a 4-celled embryonic stage. Okay, so this is the normal development they have shown, where 4 blastomeres are there, and here X blastomere is not there, is missing. All right, so if the normal development is there in presence of X, you can see Y, this is a cell lineage of Y, towards the anterior and towards the posterior. The free ends are the number of cells. They are one cell. Okay. So how many free ends do you see here? 1, 2, 3, 4, 5, 6, 7, 8. So YA will form 8 cells. and YP will form 4 cells. Okay, the free ends are the 1 cell. But when you can see X blastomere is not there, Y is forming, YA is also forming 4 cells. Actually, it won't be YA anymore because this is same as that of the YP. So both of them are forming YP. Okay. So clearly we can understand that this X has some role to play for the specification of the YA. So in a hypothetical organism as at a 4-celled embryonic stage, blastomere X instructs one of the daughter cells of an adjoining blastomere Y to take the cell fate YA. The other daughter cell takes the fate YP. This is illustrated in the figure. If the X blastomere is removed, both daughter cells take up the YP fate. In that case, YA is not there. This instruction is mediated by a paracrine factor which is called as PAP2. So they are saying that X releases a factor which is called as PAP2, and they interact with another factor present in Y which is PAP5. So this is the receptor probably, and this is the signal, diffusible signal. So PAP5 being a receptor, it will be present on the membrane of Y, right? The following experimental manipulations were carried out, which involved creating partially genetically mosaic embryos in vitro and following the fate of Y blastomere. Your question is which one of the mosaic will show a developmental pattern similar to that when blastomere is removed. So each of this is one mosaic, and you have to answer which out of this will give a pattern same as this experiment where the X has been removed. Okay. So see, the X blastomere nil for PAP2. Null for PAP2 means even if X is present, let's say X is present, but it is not releasing the PAP2 signal. Okay. So even if Y is wild type, even if Y is wild type, but it has the receptor that is PAP5, but if signal is not there, then the YA cannot form, no. So in that case here also there will be no signal. So YP, this will generate a similar one. So option 1 is the right answer. I'll justify why others are not correct. Wild type X blastomere. So if you have wild type X blastomere means it will release out the PAP2, and it is constitutively activated in PAP5 blastomere. Okay. So Y blastomere. So PAP5 in Y blastomere is always activated. So whether the signal is there or not doesn't matter. Y has the receptor always activated even without the signal. Okay. So that is why it will give rise to the anterior, and in that case all the cells will form anterior. So in that case it will form both anterior and anterior parts. Okay. So this will not give both posterior, this will give both anterior because always the PAP5 is activated, and because of the PAP2 PAP5 signaling, the Y cells will become anterior. So that's why second is not the same as this one. Then X blastomere null for PAP2 and constitutively activated PAP5. So here they have taken X as the first one, PAP2 is not there. But how does it matter if PAP5, if Y still has PAP5 activated? So PAP5 being present, the receptor being present is one thing, and the receptor always activated is another thing. Okay, so constitutively activated means it's always active. So even if the PAP2 is not there, doesn't matter. Still, Y will be able to make YA because PAP5 is activated. So it won't be the same as this experiment. X blastomere null for PAP5. So first of all, X blastomere doesn't need PAP5. It only needs to have PAP2 so that it can give the signal to Y. So if it is nil for PAP5, doesn't matter. It will still be able to give the signal to Y, and YA will be able to form. Okay, because the Y is wild type, so it will have PAP5. So PAP2, as long as it is there in X blastomere, it will be able to signal the Y, because of which Y will be able to form YA. So in all these cases, YA will definitely form, and in some of them an extra set of YA can also form. Okay, so that is not matching with this experiment. Only the first experiment where the X doesn't have PAP2 but Y has PAP5 present. So without the signal, PAP5 won't get activated. So in that case, there is no anterior, only the posterior will be formed. So correct option is option 1. Okay. So that's all from my end. All the best, students. I sincerely believe that Biotecnika has helped you a lot in your preparation, because practicing question is an essential part of CSI preparation. And in case you feel that you want to clear the next exam, then I encourage you all to join our ongoing batch. And very soon, a new batch is also going to start. You must join that, so that the next exam you can definitely clear it, even if this exam doesn't go well for you. Okay. So thank you everyone. That's all from my end. All the best.